Hello AP Bio. So here's a little video of the origami organelles of the electron transport chain, which I also added a big orange mitochondria into the background, and then the ATP synthase we made in class. So I wanted to hold this and zoom out so you can see it all set up, and then I'm going to put it in my iPad stand, which actually can't show the whole thing all at once. Alright, so here we go. Quick walkthrough showing your 3D version. By the time we are here at the electron transport chain in the inner mitochondrial membrane of the mitochondrion, we have already done glycolysis and broken glucose in half and made a little bit of NADH from NAD. We have already done the Krebs cycle in the mitochondria as well, and not only made more NADH, we've also made some, oops, I forgot to add hydrogens here. There we go, FADH2. These electron carriers are the reactants that start off oxidative phosphorylation, and as we come over here, oxygen is our other important reactant in making this whole thing happen. So we start with NAD, and I'm going to do the simplified version of this because we got all these different electron carriers and things like that. But basically, NAD holds onto just one hydrogen actually, and 2 electrons. Okay? So for every NAD, and there's lots of these, right? There's 12 hydrogens just in one glucose, and this is happening on many glucose molecules at once. As it comes close here to complex 1, which has a bunch of different proteins in it that we talked about in our other video, the electrons are going to feed through. Here we go. This is fun, right? They're going to go inside here into our electron transport chain, into our pathway. And that is going to provide the energy for protons to go through. Oh, it got stuck. Pretend it got look through. Ta-da! They got through to the other side. All right. So this happens over and over again because there's not just one NADH, but there's a bunch of them. So more and more hydrogens go across, and more electrons shuttle through here. Let's see if we can get them out of here. Well, they're in there. They're going to keep going. All right. Now we have FADH2. 2 hydrogens, 2 electrons, and similarly, electrons are going to get brought, and we're going to stick them in. There you go, like putting a little ticket in the taker. And as that happens, we have more proteins going through. Protons, woo! No, I said proteins, I meant protons, woo! They can go through here. There's all these different little proteins, woo! But basically, as we drop off, NAD returns back to NAD, and this is going to go get recharged up again, and it's going to come back with more electrons and more hydrogens that can do the same thing, and FAD's going to go back to the Krebs cycle and get charged up again and do the same thing, and eventually we're going to have tons and tons of hydrogens built up on this side of the inner mitochondrial membrane. So we moved them from the matrix to the intermembrane space, and these electrons just kind of pass along from one complex and protein in this pathway embedded in this phospholipid bilayer to the next. Okay. Now, in this last step over here, I'm going to actually talk about ATP synthase first, and then come back to how oxygen plays a super important role. Okay. So the whole point of doing all this was to build up all these hydrogens, and that is for ATP synthase. So you have this one already built at home. We did this in class. I'm going to give you the option to make this one, or you can just watch this video and look at mine. If you're a hands-on person, you can do this at home and play around with it. Here is ATP synthase, all right? And so it goes this direction. So this blue part is going to face the intermembrane space. This green part faces the matrix, and it has some ADP and phosphate chilling kind of near or in this bottom part. And once hydrogen builds up really high, hopefully we always just have tons of hydrogen because we have plenty of glucose available to us at all times in the intermembrane space. These hydrogens, and actually the one that came with this one, my cat chewed on this one, was green. So it's going to go through this peripheral stalk, and it's actually going to hook on to this little hook, and this provides energy to spin it around. It actually spins, and I'm doing a terrible job of that. Spins. And as it spins, it's then going to hook on this lower one, and in the process of it spinning, has a lot of power, a lot of energy as it's spinning. It's going to help slam ADP and phosphate into each other. They're drawn towards each other already. Just puts them right into contact to form ATP, and now we have ATP in the matrix. But the problem is, now we have hydrogens coming through. And so we don't want hydrogens to build up on this side now, because then there won't be more hydrogens on this side than on this side. We won't have high, low. It'll just be equal, and ATP synthase will stop. We don't want ATP synthase to stop, because then if that happens for too long, you die, because you can't do anything without ATP. So, handy dandy oxygen. This is why we say oxygen is required for this. These hydrogens, along with, that's why I said I'll pause and come back to it, the electrons that were at the end of the electron transport chain, these electrons need to go somewhere. They can't just like chill here. They're bouncing around. They have lots of energy. We use the energy in these electrons to get over here. We split them apart. These electrons, all right, need somewhere to go. These hydrogens need something to do as well. So as the hydrogens flow down back through ATP synthase into the matrix, 2 hydrogens and 2 electrons. Let's see if I can find another one in my electron transport chain. There we go. I'll try to shuttle it. I'll just grab it from here, but pretend it went through there. Okay. Our 2 electrons, negative, negative. Our 2 hydrogen ions, positive, positive, react with 1 oxygen, and we combine all these together. What do we get? We get water. And I'll draw that on a little post-it. Ta-da! Water. And so water, as you know, is one of the products of cellular respiration. This water is not the reason we do it, so it can just stay in the cell if it's needed there, or go do whatever it needs to do in the body, or you can sweat it out, pee it out. Your body knows what to do with water. It's not toxic. And we have another oxygen here, so as more hydrogens come across through ATP synthase, it's spinning around, woo, to make more ATP, which I dropped on the floor. Yay, ATP! As it makes more ATP, we have more hydrogens build up, so we need constantly more oxygen. We have more electrons flowing because we're making lots and lots of energy every second, every moment of your life, and every cell, and we make another water molecule. Okay? So that is the little 3D model that I hope also helps you understand this last step of cellular respiration, which is called oxidative phosphorylation. It involves the electron transport chain and then the process of hydrogen building up high and moving to low through ATP synthase to make ATPs called chemiosmosis.